After eversion and the process of convergent extension and isotropic expansion, at about 7 hours after pupae, the blade starts to swell because there's fluid entering into it, and the two epithelial layers, the dorsal and the ventral one, get separated. At this time, a cuticle starts being secreted by the epithelium. So at 18 hours after pupae, more or less, this cuticle is already secreted and the blade is attached to it. And also both epithelial layers become in contact again. But this time they leave these holes in the middle, which are going to be the veins. So these cavities are left where the veins are going to be. At this time, at about 16 or 18 hours after pupae, the hinge starts contracting. So it gets cells get smaller, and this contraction is asymmetrical in such a way that the anterior part contracts less than the posterior part. This contraction pulls from the blade, and the blade gets deformed, so its shape changes and it becomes very similar to the adult form, which is asymmetrical in the anterior-posterior direction. Here you have a video of the whole process. See how here the hinge is contracting and is pulling from the blade, and now veins begin to differentiate. So the vein territories were already specified, but they hadn't differentiated. If you see at the beginning of the process, there are a lot of these bubbles, which are cell divisions. So cells first increase in size and then divide. As you have already seen, the main forces affecting wing shape at this stage are global forces, mainly the attachment of the wing blade to the cuticle and the hinge contraction. Attachment of the wing to the cuticle is through this protein called DAMPI, which we have already mentioned. It is a very big protein of about 2.5 megadaltons, and its locus is also big, about 100 kilobases. In Drosophila, there are other loci of this size, but they are mostly non-coding, meaning that they have lots of intron, etc. But this protein, in this locus, most of the sequence is coding, so it is particular in that way. It is a highly modular protein. It has more than 300 EGF repeats, which are these green squares, and they are interspersed with these pink DAMPI modules, which are called DAMPI because they were discovered in this protein. Here's DAMPI expression. As you can see, at 18 hours after pupae, it is expressed mostly in the border of the blade, which is where the blade is attached to a cuticle, although it is also expressed in the surface of the wing, mainly at later stages. Here you can see how it is expressed in the border of the blade. Different DAMPI mutants have different phenotypes, such as these oblique phenotypes, which is very mild, and the wing has a different shape, but it is mostly well formed, and these truncate phenotypes that are more conspicuous. Then in E, you can see the effect of repressing DAMPI expression with RNAi in the whole wing using a nubin promoter, and in the F, you can see the result of repressing DAMPI expression only in the border of the wing with using a distalless promoter, and as you can see, the phenotype is very similar. If you repress DAMPI in specific patterns, you get predictable phenotypes. As you can see, if you repress it between veins 3 and 4, just in the middle, you get a little bit of invagination in the middle part of the wing. If you repress DAMPI in a wider area, then you get more invagination, and then the borders. If you repress DAMPI in the borders, you get more elongated or less a wing that is less tall. So these phenotypes are predictable. Here you see a control and a dumpy mutant wing, and as you can see in dumpy mutant, since the blade is not attached to the cuticle, when the hinge contracts, the wing blade goes after is pulled by the hinge, and it detaches from the most distal part. Here's the proof that the dumpy function is mechanical, because it's the phenotype can be mimicked by just cutting the wing blade with a laser. Here the cut is performed at 20 hours after pupae, and the picture is taken at 32 hours after the pupae. So as you can see, if you cut the blade at different places, the retraction has a different pattern, which is very similar to the patterns of the dumpy mutants. So the dumpy function is mechanical. In the leg and also in the antenna, dumpy is also expressed at the tip, so probably it has a very similar function in these appendices. Here you see the phenotype of repressing dumpy in a leg, which gets shorter, and in an antenna, which also gets shorter, as you can see here, signaled by the arrow. On the other hand, we don't know much about how hinge contraction is regulated, and what we think we know is mostly indirect and it comes from the observation that the cells that are in the veins also decrease their size during this stage. There's a transcription factor called SRF or MAL, which is expressed in the blade intervein region, and also, but it is not expressed in the veins or in the hinge. So when it is overexpressed in the vein, it causes a little bit of overgrowth, and veins do not, the size of cells in veins does not decrease. And as you can see here, in a wild-type fly, vein number 5 exerts some tension, so that the border is somehow a little bit invaginated here. But in this, if you overexpress SRF, this doesn't happen. So the hypothesis is that the lack of this transition factor expression leads to cell contraction somehow. During this process, the blade tissue is shearing, but this shearing is not homogeneous in time and space, so it can be measured. So here you can see how the shearing is oriented at 17 hours, 30 minutes after pupae, at 21 hours, and at 26 hours, where shearing has mostly stopped in the most distal part of the wing, and it's still significant in the most proximal part. The contributions of different cell behaviors to this total shearing can be studied. So let's begin in the lower part, which is the lower part shows the contribution of cell division to total shear. As you can see, most shearing due to cell division takes place at the beginning of the hinge contraction process, between 17 and 21 hours after pupae, whereas at 26 hours there's no cell division anymore. Then there's a contribution to shearing that is mediated by cell elongation. As you can see, this is more important at the beginning, whereas in the end, cell elongation goes in the opposite side. So mostly because cells are at the beginning, cells get elongated in the proximal distal axis, and then they become more relaxed. So they start being more rounded again. And finally, you have this shearing that is due to T1 transitions, which are these, as already explained, these changes in cell topology, in the neighboring relationships between cells. And as you can see, at the beginning there are lots of T1 transitions, but they are not oriented in the proximal distal axis. However, at the end, they are oriented they become oriented in the proximodistal axis. Here you can see the aggregated data. This blue line represents the total share, which is higher at the beginning and then it slowly decreases. This green line represents the total shearing, the cell elongation, which, as you see, is much higher at the beginning, and then it becomes negative. So if it becomes negative, it means that it is in the vertical direction, in the anterior-posterior. It's anterior-posteriorly oriented, whereas positive values mean proximodistally oriented. So this is the pattern of the cell elongation, and the pattern of the T1 transitions is exactly the opposite. So what's happening is that here there's a peak of oriented T1 transitions that are oriented in the proximodistal axis, and this is actually what is allowing these cells to relax and become more rounded, which means less elongated. So basically these in the effect on the total share is that T1 transitions are compensating for this cell relaxation, or vice versa. Intriguingly, at the beginning of hinge contraction, there is a negative contribution of T1 transition. So here's the thing with T1 transition. T1 transitions are normally triggered by sharing, by the tension that cells that are being pulled. So when these cells in the horizontal plane are being pulled, their interface becomes shorter, and eventually it is replaced by another interface. So it is a passive process. However, if T1 transitions are taking place in the opposite direction of the tension, it means that there's probably some active behavior of the cells that so cells are making this transition because they want to somehow, rather than because they are under tension. So this is this is interesting. This would be the same time of plot, but for a dampy wing. As you can see, total shear is much lower compared to the wall type, which is in the corner. T1 transitions in the beginning have a negative contribution to shearing, but after 24 hours there is not this increase in the shear rate due to T1 transitions, like as it happens in the wall type. And in the same way, cell elongation, which is the green line, is much milder. After we have determined which cell behaviors take place during this process, we may wonder which specific cellular mechanisms or more molecular mechanisms contribute to alleviate this tension. So in this paper, they perform again this kind of laser ablation experiments in which they cut cell junctions through the red line, and they measure the retraction velocity in the blue spots. So they see how in the upper row you see the junctions before laser ablation, and in the lower row you see the junctions 10 seconds after ablation. As you can see, 16 hours after pupae, there is not a lot of retraction, so cells are not under a great tension. But at 20 hours after pupae, there's a lot of retraction, so cells here are under a lot of tension. And then at 32 hours, this tension gets lower again. Here you can see the pattern of how tension, or measured as the recoil velocity, increases at 20 hours after pupae, and then it steadily decreases until 32 hours because this tension is getting relaxed. However, if you compare this to cell elongation, you see that the peak of cell elongation is a little bit out of phase with the peak in mechanical stress. And this is interesting because if you pull from an elastic material such as a rubber band, you expect that the length of the material is proportional to the force or to the tension, or to the stress that you are applying on it. So if you stop applying this force, the elastic material will immediately become shorter. So however, in viscoelastic materials, when you stop pulling from a viscoelastic material, there's a delay, and it triggers a while before it can get shorter again. So when you put the stress for the first time, its length increases, but when you stop this stress, it triggers a while to return to its original position. So when they plot the recoil velocity, which is the tension, with cell elongation, they get this kind of shape, meaning that the wing tissue has this viscoelasticity, and the area inside of the circle is related to the amount of to how much viscoelastic the tissue is. So which molecular mechanisms are responsible for this viscoelasticity? Well, it was already known that E-cadherin, which is an element of adherence junctions, which we already mentioned, so the turnover of E-cadherin in cell junctions is modified under tension. So they build a reporter of E-cadherin, like they tagged E-cadherin with GFP, and they perform these FRAP assays, which are these are assays that are based on the phenomenon of photobleaching. So when you put under a laser these fluorescent proteins, these proteins lose their fluorescence. This is what is called photobleaching, but the molecules are still there. However, molecules that are outside of the laser do not lose their fluorescence. So after a while, molecules move around, and molecules that are not fluorescent are replaced by fluorescent molecules, and you see a recovery in fluorescence. So the time of this recovery is proportional to the turnover rate. So as you can see here, the fluorescence is recovered quite fast in a wild-type wing. However, in a dampy wing, which is not under pressure, which is not under tension, the fluorescence is not recovered, meaning that tension is affecting the turnover rate of ECAD. Here you can see the recovery curve in the wild-type and in the dampy, which is much the time is recovery is much slower. And the same happens after laser ablation. Instead of having a dampy mutant, you just cut the wing from the margin, and you get the same result because there's no tension. So ECAD turnover is regulated by tension. Then they find another player, which is P120. P120, when there's no tension, under circumstances in which there is no tension, is bound to the cell membrane, and it binds to ECAD and prevents its turnover. So it prevents that ECAD is endocytosed in these vesicles. So it decreases the turnover rate. So when they measure this would be a wild type again, and then dampy mutant. So the dampy mutant has a lower recovery rate. But if they measure recovery rate in a P120 mutant and a double mutant, they see how in the double mutant the recovery rate is the same. So the effect of dampy is reversed, because although there is no tension in these junctions, although there is no tension in dampy mutant, P120 is not there to repress the turnover or to decrease the rate of turnover of E-cadherin. So P120 has a role in regulating this viscoelastic property. Indeed, as you see in this plot, the area inside of the curve is much smaller than in the wild type. Inside of the curve of the P120 mutant is lower than in the wild type, which means that the viscoelastic properties of the wing are reduced. And also, the final shape of the wing is changed. It has an increased size, as you can see here, and it is more elongated, which possibly means that regulating these mechanical properties, the viscoelastic properties of the wing, you can modulate the adult shape of the wing, but of course you cannot be sure that P120 is affecting shape by other means. Finally, DPP is also required for growth and cell division in the last pupal stages. In this work, the authors repress DPP expression 24 hours before pupae, and here they show that DPP signaling in the wing disc is normal as assessed by PMAD staining in the wing disc prior to downregulation of DPP. Therefore, they are not affecting the growth of the disc, and they are not affecting final size of the disc or cell division in the disc. They are specifically affecting DPP expression in the pupal wing disc, in the pupal wing. So they find, as you can see in this quantification, that the final size of the wings when DPP has been downregulated is lower than in the wild type. And additionally, they show how cell division is also decreased in the mutant compared to the wild type. Cell division here is assessed by staining of phosphohistone 3. Changes in the dynamics of all these processes can result in variation in the shape of the adult wing. Obviously, dynamics of hinge contraction are very important. For instance, how much cells contract, or when they start and they end contraction, or how much asymmetric this contraction is with respect to the anterior-posterior axis. Also, the pattern of adhesion of the blade to the cuticle might be important, and this can be related to changes in dampy protein expression. Also, changes in tensile and adhesive forces between the cells, meaning that the changes in the polarization of the actomyosin network or DATs, etc., can result in changes in the pattern of T1 transitions or in the cell shape and the viscoelastic properties of the cells. Then dynamics of vein differentiation could also be important, because there's a difference in tension between in the vein and intervein junctions. So veins have like a higher tension and they pull on the wing margin. It is easily seen in the end of vein 5 that there is like a small invagination of the margin, and this can be the result of this tension that the veins have. Finally, changes in the gradient of DPP, or on the amount of DPP that is expressed in the veins, can result in changes in the final size of the wing and changes in the pattern of cell division. The final stages of wing development are not understood in detail, but it is thought that they do not contribute much to the final shape of the wing. Between 46 hours and 96 hours after pupae, the adult cuticle is secreted. Here it is represented as this black line, and the wing grows without cell division, which means that it grows mostly due to increase in cell size. So it grows and it bends inside of the cuticle. Then after closure, most of the cells in the epithelium migrate towards the body, leaving only a few neural cells in the veins. Finally, I will leave you with a video of how the wing expands after a closure.